thank you for tuning in for the 5th episode of the Doc Talk with Liz podcast. On this week's episode, our guest star is Dr. Kayla Davis, who just recently graduated with her PhD in neuro and cell biology from Harvard and is currently working as a postdoctoral researcher at Boston Children's Hospital. I hope you enjoy. Hello, Dr. Davis. Thank you for joining me today.
Hi, Lizzie. It's great to be here.
I just was hoping you could start off by telling me about your journey into science, kind of like what led you to where you are now.
Sure. Yeah. So I, first realized that I was interested in science in high school. In high school, science kind of started to diverge for me away from some of my other classes because it was kind of hard. I started taking these like advanced biology and advanced chemistry, and I felt that I was really having to think a little bit harder about my homework compared to some of my other classes where I was like, memorizing a lot of material and repeating it, like Spanish class or history class. But science was more challenging for me. And so I really enjoyed that aspect of it. I started kind of thinking, what do I want to be when I get older? And I said, I want to be a doctor. I want to be a medical doctor. And I started looking into that. And around that same time, I had a really great high school biology teacher, Mrs. McKee. And she took us to a conference that was designed for high school students, where a scientist traveled the country and he gave genetics lectures to high school students. And this kind of opened my eyes because he started talking about, the current genetics research and the potential future of genetics research. And that just like totally fascinated me that this could even be a thing. And so I'm still thinking, medicine at this time, mostly because all I'd really been familiar with, I grew up in Stillwater, Oklahoma, and all I was really familiar with growing up there was, if you like science, you become a doctor. That's your pathway. And so I started interning at the local hospital. I would volunteer there and help patients, and I would intern and I would shadow doctors. I shadowed an anesthesiologist there, and I thought this is really cool. And around that same time, I applied for a summer internship where you get to shadow doctors. And I was competing against some of my really fantastic classmates, and I actually didn't get the internship. And I was just like so crushed, like, I want to be a doctor. This for me was my pathway to doing this. And so I was talking to, it was my physics teacher at the time, Mr. West. And he said, I met this scientist the other day at the local university, Oklahoma State University. She's a biochemist and she's looking for a high school student in her lab. Are you interested? And he gave me her contact information. Wow. And I was really brave, and I just picked up the phone and I called her because I didn't know any better at the time. I just called her and I said, my name is Kayla. I'm at the time I was in the 10th grade. And, I'd applied for this internship for the summer. I'm really interested in medicine. What do you have going on? And so she said, come in for a day at the lab, meet me. And so I came in and I met her. Her name is Dr. Rita Miller. And she, kind of told me about her lab. She was really interested in the topic of cell division. And she explained to me why studying cell division is so interesting and important, and how cell division and issues of cell division can underlie a lot of different diseases, and particularly cancer. And this really caught my interest. I said, whoa, I could study things that are important for cancer. And as this, sophomore in high school, and that caught my attention. And so I said, okay, I'll sign on. I'll stay with you for the summer. And so I stayed with her for the summer. And a lot of what I did was just kind of like data entry and the computer. And I would shadow if someone was doing an experiment that day, and I would go to lab meetings. But this experience really opened my eyes, and it made me kind of realize, yeah, I'm really interested in science. I'm really interested in medicine. But the part that I like about science and medicine is actually thinking about and understanding the diseases, and not so much the previous experiences I had done where I was shadowing the doctors and interacting with the patients. So that really opened my eyes. And for the first time in my life, really, I had this example of a female scientist sitting in front of me who was really invested in training me. And that just totally changed everything for me. I decided that, I really did want to be a scientist from there on out. And I continued to work with Dr. Miller. I came back for the summer after my junior year of high school. Dr. Miller helped me figure out where I wanted to apply for college, what I wanted to study. And I ended up deciding to stay at Oklahoma State University for my undergraduate career. And then I worked with her through my entire undergraduate career as well. And so she really fostered my interest as a scientist and, showed me all these awesome things and how I could, take this information and go off and turn it into a career.
Awesome. So I was also going to ask you who some of your scientific role models are, and I'm guessing Dr. Miller is one of them, but are there others?
Yes, so Dr. Miller was definitely one of my role models. I mean, and she just set such an amazing example for training students. And, after I came through her lab, she's continued to take students, particularly through a program at Oklahoma State University called the LSAMP program. There are LSAMP programs at a lot of universities for underrepresented minorities in science, where they'll pay you a stipend to work in a lab as an undergraduate. She takes a lot of these students and trains them and helps them figure out, how to go forward in their career path past undergraduate. So she's definitely a role model. Other role models I have are scientists who I think have really combined kind of their love and passion for activism with their science. And so one of those role models is a scientist that I've been very lucky to work with during my graduate career, named Dr. John Beckwith. He started a course called Social Issues in Biology, which takes really a deep dive into all kinds of social issues that affect scientists, like racism and sexism and social class. And we talk about how this, intertwines with medicine and the science that we're allowed to do and how people perceive our science. And so Dr. Beckwith has been a huge role model for me in that sense. Another role model lately has been Dr. Kizzy Corbet, who's all over the news for her amazing research with the mRNA vaccines. And she was part of the group at the NIH that helped develop the Moderna vaccine. And, one thing that I really respect about her is how much outreach she's doing. A lot of times scientists will do this incredible work and they kind of, hide out in their lab and they don't really interface with the public. And she is, on the nightly news, she's going to churches, she's talking to people, and she's doing incredible outreach work to talk to people about the safety of vaccines, but also to put a face to these vaccines and kind of, humanize the whole experience of being a scientist. And I really respect what she's doing for the entire scientific community in that respect.
Absolutely. And then also going back to, you commented on cell division and cancer. Do you think that you could kind of explain that a little bit more? So cancer is kind of caused by out of control cell division?
Yeah, so whenever you have a cell, so most of our cells, we call somatic cells, they divide. And so what happens when cells divide is all of the DNA in cells have to align in a particular way so that when they divide, so that when they copy and they divide, the DNA stays with the mother cell, but this copy of the DNA also goes into this daughter cell, which divides. And you want it to be as close of a copy as you can to the mother cell, right? So it's really important that these things actually physically align in a way so when the cell starts to divide and pulls apart, everything is lined up and on the right side of the cell that it needs to be. When in issues like cancer, a lot of times there are issues with cell division where the DNA or different pieces of the cell will not exactly align the way that they're supposed to be. And this process will not happen the normal way. And that is when cell division can kind of get out of control. And you can have these cells that, have the wrong set of DNA or information in them, maybe too much DNA or information in them. And that can start to cause problems in our body.
Okay, awesome. Thank you for explaining that. And then I was also wondering, so you said that you decided to stay at Oklahoma State University for your undergraduate. What kind of undergraduate research did you do that also kind of like really showed you I definitely want to be a scientist on top of what you had already experienced?
Yeah, so, I'm going to backtrack a little bit because whenever I was applying to colleges, I was looking at places, that had genetics type degree programs, and I was applying to places in California and Colorado, Oklahoma, all over. And, the reason that I ultimately decided Oklahoma State University was because they had scholarship programs for undergraduates in research. So there was one called the Freshman Research Scholarship that I got, which put you in a research specific class for freshmen, helped you find a lab to work in, helped you kind of work on putting together a CV and contacting professors to work with and that kind of thing. And then they had several other programs that you could take advantage of throughout your undergraduate career where they would actually pay you a stipend to do research in a lab full time. And so for me, that meant that I could make my job working in a lab and get paid to do that. And that could help me pay for my tuition and fees instead of having to find a job outside of school. And so that opportunity just really stood out as significant for me. And so that's why I decided to go to Oklahoma State. So while I was there, I worked in Dr. Miller's lab throughout the school year. And then during the summer, I would apply for summer research internships. And so one of my summers, I did stay at Oklahoma State University and I worked with Dr. Miller, and that was part of my fellowship that I would stay there throughout the summer and work full time as well. My dog's going up the stairs. And he wants to join. And during the other summers, I applied for what's called a research experience for undergraduate. And so there are a ton of these experiences. They're funded usually by the National Science Foundation or the National Institutes of Health. Almost every institution has at least one. And they bring in undergraduates to work on campus during the summer. And many of them will house you for the summer on campus in a dorm. They'll pay you a stipend. They will feed you many of your meals. They'll fly you out and back home. And so this was just an incredible opportunity to try to do research somewhere else. So I applied after my freshman year of college to probably 10 programs and ended up getting accepted to a program at Johns Hopkins University, their Bio-REU program. And I spent a summer at Johns Hopkins, I think it was 10 weeks, working in a virology lab that studies RNA viruses. Very important now. I didn't even understand when I was there exactly how important these things were. And then the summer after my junior year of high school or junior year of college, I applied for summer internships again, and I applied and ended up at a program at Harvard Medical School called SHRP. And that was a fantastic program. They housed us on the Harvard Medical School campus. I got to work in a lab with Dr. Wade Harper for 10 weeks studying cell biology. And that experience was really big for me. I ended up applying to Harvard for graduate school because I enjoyed that experience so much.
Wow, awesome, awesome. And so could you also talk a little bit about so I know that you just graduated with your PhD, congratulations. And so could you kind of tell us a little bit about what you research now, or what you got your PhD in and that kind of thing?
Sure. Yeah. So when I applied to graduate school, I applied to the Biological and Biomedical Sciences program at Harvard University, or we call BBS. And BBS is an umbrella program, which a lot of universities have, these big programs where, there's kind of just a broad topic like biomedical sciences. And there are a lot of different types of scientists that you can work in under that umbrella. So as a student in the BBS program, there were like 500 labs that I could possibly work in, which is just incredible, right?
Yeah.
And so I was really interested. I wasn't exactly sure what I wanted to do when I applied to graduate school. And so I kind of focused on programs like this where I would have a lot of options and I would have a really broad education as well, instead of something like very narrow and specific, like just a neuroscience program, for example. But when I came in to graduate school, I'd already, kind of worked in a cell biology lab at Oklahoma State University on cell biology and biochemistry. When I worked with Dr. Wade Harper over the summer, I'd worked in the cell biology department. And so I knew I really liked that type of science. And so just that level of science where you're looking at cells, you're looking at things inside cells and how they communicate with each other. And so I joined a lab in the neuro department at Harvard Medical School and Boston Children's Hospital. And it's Dr. Thomas Schwar's lab. And it's turned into kind of a neuronal cell biology lab. So we study cell biology questions, but we like to use neurons to ask those questions. And we're really curious about how these cell biology questions particularly relate to neurons and neurological diseases. And so we and the Schwar's lab are very interested in mitochondria specifically, and I've worn my mitochondria earrings to talk about it. Thank you. And so the reason we're really interested in mitochondria is because they are so important for cell and specifically neuronal cell health. And so I'll just back up a little second. When I say neuronal cell, I'm talking about the neurons that make up our brain. So neurons are the specific cell type that make up our brain. One reason that mitochondria are so important in the neuron, we think about mitochondria, you probably learned in your biology class in school, they're the powerhouse of the cell, right? That's what we all remember about mitochondria. The reason we think about them as the powerhouses of the cell is because they literally make all the energy that our cells need to survive. But whenever we think about mitochondria, we think of them as kind of like these little bean-shaped things. They sit in your cell. Yeah, they provide energy, but it turns out they actually move around. And so if you watch them in a cell that's alive, they wiggle and squiggle all around the cell. And the reason they do that is because it's really important that they get to the areas of the cell where they're most needed for providing energy or performing other functions that they have, like buffering calcium within cells. Mm-hmm. And so in normal cells, they move around and it's not that big of a task for them to move around. But in neurons, it can become really, really important because our neurons can actually get quite big. So some of our neurons in our body can be up to 1 meter long in length, which is an incredible length, right? Mm-hmm. So you're thinking about these tiny little mitochondria organelles, and they're tasked with this job of moving these really great distances, several hundred micrometers or up to 1 meter in length. And so these little mitochondria actually have little motor proteins on them. Maybe you've seen sometimes there are like cool YouTube videos and stuff of these motor proteins, and they walk on microtubules, which are in our cells, and microtubules kind of serve as these railways. And then you have these motor proteins that they actually like walk along these microtubules. And so these motor proteins sit on these mitochondria and they physically walk and move them to the areas of the cell where they need to be. Mm-hmm. So what's really interesting about this is whenever your cells are healthy, your mitochondria are moving around, they're going everywhere that they need to be. But what we're really interested in is what happens when our mitochondria aren't moving around the way that they're supposed to normally anymore. And so we know that as people get older, our mitochondria actually start to slow down. They stop moving as fast as they did when we were younger. And we know that in certain diseases, like Parkinson's disease and certain types of Parkinson's disease and certain types of dementia, these can actually be caused by issues with our mitochondria, where they're not functioning as properly and they're not moving the way that they should. And so whenever they're not moving the way that they should and the mitochondria can't move throughout these neurons, the neurons will actually start to degenerate. They will get shorter and shorter. And the problem with that when they degenerate is normally our neurons are stretched out. They have these synapses. These synapses connect one neuron to another neuron. And that's how they send signals from one cell to another cell. Like if I say, I want to pick up a glass of water. Right now, my neurons are firing, my synapses are firing, and they're telling my brain how to do this, right? So I can pick up my glass of water. If my neurons aren't in close proximity and they can't talk to each other anymore, then it might be hard for that signal to be relayed, right? Mm-hmm. And so it's really, really important that these mitochondria function the way that they're supposed to function so that these neurons can be healthy and they can talk to each other and our bodies will work the way that we want them to. Mm-hmm. So we are very interested in all the ways that mitochondria move, all the ways that mitochondria movement can be regulated in these neurons. And we try to find new ways that these mitochondria are regulated so that we can better understand how these regulations might relate to different types of neurological disease.
Okay. To recap, in the first part of this podcast interview, Dr. Davis mentioned that she was connected with a researcher at a university near her who she was able to conduct research with while in high school. She also mentioned that there are many different avenues for studying diseases and that science can be an alternative route to medicine. When cells divide, the DNA has to align in a particular way so that the DNA remains with the mother cell and goes to the daughter cell. REUs, or research experiences for undergraduates, can serve as great opportunities for undergrads to explore scientific research and other colleges while earning a stipend. Neurons are the cell type that make up our brain. Mitochondria make all of the energy that our cells need to survive. And lastly, mitochondria have motor proteins that walk along microtubules to get to the part of the cell where they need to be.
Okay, awesome. And kind of going off what you said, so I'm kind of wondering about some of the biochemistry behind this. So like what is that energy called that the mitochondria produce for us, and how do they like actually produce that energy? Like, do they need an input for that specific output, or kind of what's the biochemistry behind that?
Great question. So in your college biology class, you probably learn about ATP, and ATP is this energy molecule that we need in our cells, right? Mm-hmm. And our mitochondria pump out tons and tons and tons of ATP. They can take basic molecules, bring them into the mitochondria, break them down, turn them into ATP, and pump this out. And so they kind of like work as little powerhouses. They literally cycle these molecules over to make this energy. And they can transform this molecule ADP into ATP, which can work as energy.
Awesome. Thank you. And then kind of transitioning a little bit, could you kind of give us a glimpse into the life of a scientist? You know, every single day is different, but different things like that you do, and also ways that you can combine your other hobbies or interests with your like career in science.
Yeah. So my regular life, so I you know, being a graduate student in science is kind of like having a 9-to-5 job plus a little extra. So I'll just give you a little overview of what kind of my graduate school breakdown was like, because it changes as you go through. So at least in my program, our first 2 years of school, we take classes. So we spend a lot of time taking classes in the morning, and then you'll kind of go to your laboratory in the afternoon. And then after our second year, we transition to just being in the lab pretty much all the time, unless you're doing like teaching as well. So I, you know, come into the lab. Sometimes some scientists are kind of well known for liking to sleep in a little bit. And so we usually start around 10 a.m. We all, you know, come to lab. Usually what I do, I'll plan out my week about a week ahead of time. I'll say, you know, these are the experiments or the questions that I really want to focus on next week. This is kind of the rough schedule that I'm going to do them on. And then as scientists, we actually have to be really flexible because a lot of times science doesn't go as planned. And so you write out this ideal schedule. This is what I'm going to do this week. And then you get to the lab on day one and your cells didn't grow as fast as you want to. And okay, I need to push that till tomorrow. My whole day gets pushed. So anyways, we come in, the type of things that I do on a day-to-day basis. So I do a lot of pipetting. So I have my pipette and I do things, a lot of like molecular biology. So we clone plasmid constructs that we want to use in the lab. So that's a big, I'll break that down for a second. So a plasmid is a vector which you can actually put like a gene of interest in. So say I have a gene of interest that's on mitochondria. My favorite gene or protein that's on mitochondria is called Miro. And so what I can do is I can actually take the DNA sequence of this Miro. I can put it into a plasmid, which is just like a string of circular DNA. And I can insert that sequence for Miro there. And then I can also insert the sequence for, say, a green fluorescent protein called GFP next to Miro. So then I have green fluorescent protein and Miro, and I can put them in this plasmid together. And whenever I take this plasmid that I've cloned together, we're kind of designing, molecular engineer these plasmids, and I can actually take that and I can insert it into live cells. And if I insert that into live cells, then it gives these cells the instructions to make this green fluorescent protein for Miro. And then it will make Miro in the cell glow green. Wow. And so this is a tool that we use a lot in molecular biology and cell biology because it allows us to actually see these proteins that we study, see where they're at in the cell. If I make Miro green, then you see all the little mitochondria in the cell light up and you can follow them around, which is super cool. And so I do a lot of that kind of thing, kind of genetically engineering these plasmids that I want to use to ask different scientific questions for cell biology. I do a lot of cell culture. So we have different kinds of cells in the lab. I use human cell lines and rat cell lines and neurons from rats in the lab. And so I keep those in the lab and I have to take care of them. And so we feed those cells every day and just kind of check on how they're doing so that we can keep using them for experiments. And then the other part of science, I do these, I think about the experiments that I want to do, I carry out the experiments, I collect data, I spend a lot of time on a microscope, a lot of microscopy, like a lot, which is actually one of my favorite things to do though. It's just beautiful, right? And the other things that I do outside of that we don't think about so much as being a scientist is, you think of these questions, you collect this data, but what we do as scientists is we share our data. And so I actually spend a lot of time also writing. I spend a lot of time making what we call figures for our data. So putting all of our data together into these pretty images so that I can present it. And so those are kind of some skill sets that you don't necessarily think that you might need as a scientist, but you actually have to focus on your writing and how to communicate the science that you do. And then, one thing I definitely didn't plan for when I got into graduate school is I kind of have to be a graphic designer sometimes too, because I'm taking these images and I'm putting them together and I'm putting together these graphs and I need color schemes that look nice in my graphs that match my pictures and all of this, because you want, whenever you write this up, for it to be very visually pleasing to whoever you're showing it to or whoever's reading your work, right? So I do a little bit of that too. And there's a lot of freedom for, people who there are some scientists who are like great artists who make these beautiful, beautiful figures and cartoons. That is not really my specialty, but I try to do it. And so, a lot of that kind of stuff too, which is not necessarily what I thought of when I was an undergrad as like, this is how I'll be spending my time as a scientist. Right. And I'll just touch on, you asked about how I kind of fit in, other things that I'm interested in as well. So, at least within my graduate program and at Harvard University, there were a lot of universities have outlets for graduate students to get involved in their community, to get involved in teaching. And those were things that I thought were really important. And so a couple of things that I've done outside of working in the lab. So I've taught a class. So I mentioned earlier that Dr. John Beckwith is one of my mentors and someone I looked up to. I taught a class with Dr. John Beckwith, the social issues in biology class, and I TA'd that class for a couple of years and then gave a couple of lectures in the class. And that was something that I really enjoyed getting to do. And kind of thinking of these, social science issues that are related to science, but are very different from the type of questions that I think about as a cell biologist day to day. And then other things I've done, gotten involved in, there are a lot of science communication outreach groups, giving public lectures. So I've taken part in that. We have a conference that we put on every year for the general public where we invite graduate students to share topics of interest with the general public in short little seminars. We do that. I've gotten very involved and interested in science policy. And so that's the intersection of how, how policy and the laws that are passed can affect or regulate the science that people are allowed to do, or funds the science that people are allowed to do, or encourages certain people to become scientists. And then two, how the science that we do can actually inform policy. And so we saw a lot of that in the last year, right, with COVID, where it became really important to listen to scientists and epidemiologists to understand how COVID worked and what the best rules were going to be for things like social distancing and schools being open. And so I'm really interested in that area and like to spend time working on those things as well.
Awesome. And so thank you for that little background, kind of telling us about what the life of a scientist is kind of like. And so now I'm wondering that maybe there are people, they're listening, they're like, well, this actually sounds like something I'm interested in. Maybe I like science, I like math, I like graphic design, all of these different things. And maybe they want to go to graduate school or get a PhD or study some type of career in STEM or get a career in STEM. So do you have any tips for those people, like what they should do? I know that you already shared about like those different programs for high schoolers and undergraduates, but I'm also wondering about things they can do on their own, like, reading scientific literature or just different things like that.
Definitely. So, especially if it can be hard, especially if you're not, like when I was in high school, I was close to a university, right? And so I was able to start working at that university during high school, but what if I hadn't been? So, there are actually a lot of really cool resources online now that were not available when I was in high school. So some of my favorite, there's a website called Lab Exchange that has launched out of Harvard's edX group, and they have some really, really cool online simulations of the type of experiments that molecular biologists and biochemists do every day. So if that is something that you're interested in, and you don't have the opportunity to, literally get your hands wet and get into lab and do this kind of thing, I highly recommend, looking for those type of resources online, looking for them. There are also, I mentioned edX already. There are all kinds of cool classes that you can take from amazing professors and universities. There's a resource called iBiology. I just really suggest, whatever it is that you're interested in, try to find the experts in those fields that really inspire you and carry on your interest. And when they say something that you're like, wow, that's really cool, or I wonder how that works more, just keep following it up and keep asking questions, because so much of learning to become a scientist is learning how to ask good questions. And really what we are is we are professional researchers, right? And so a lot of you, I'm sure, can be quite good at research. You have a question, you get online, you start looking around, you look for good sources that are reliable for that question. And so just developing that skill of figuring out how to research your question, find a good resource for it, and draw conclusions from that as well, is a really valuable skill set that you'll be able to apply to any type of STEM area that you want to go into. And another topic that I've talked to with Lizzie about previously is, the one skill set that I wish I had earlier was coding. I didn't do any coding in high school, any coding in undergraduate. And that is something that I've just continuously seen in my research and also my colleagues' research that can be incredibly helpful and important for looking at data sets and for asking different types of questions, right? And so one thing that I do is I spend a lot of time on the microscope. So when I spend a lot of time on the microscope, I take hundreds and hundreds of images. And it doesn't really I could, sit there for hundreds and hundreds of hours analyzing all of these images by hand and counting the mitochondria and looking how they move. But it's maybe not the best use of my time to do that. And so one thing that we've been able to do in my research is we've been able to develop algorithms that we can use in image analysis software to actually analyze this data for me. It's a huge time saver. It takes biases out of data analysis. So instead of me looking at it and telling you what I think every time, the algorithm is giving an output that, without my opinion involved. And so I think it can just be a really valuable skill set that you can turn into a lot of different careers, whether you want to go to graduate school or not. Yeah, Lizzie?
Could you, and I know that you also created a resource called Code Sci Lab. So could you talk about that a little bit?
Yeah. So, this recognition that I would, love to learn how to code, and some of my friends were really good at this, kind of made me realize that if I had been exposed to this idea or resources at an earlier age, it could have made a huge impact on me. And so one thing that myself and some of my friends from graduate school did is we created a website called codescilab.org. What we do on codescilab.org, we have lesson sets, and they teach the basics of coding for solving scientific problems. And so we use the Python coding language, which is a very popular language for solving scientific problems. And we teach the basics of that coding language. But the way we do it is we actually apply the lessons and the questions you're asking to scientific problem sets. And so you can immediately start to see, whenever you start taking these coding lessons, how you can apply these skill sets to solving scientific problems. And we hope that, once people, it just kind of lowers the barrier to entry for coding, which sounds kind of like big and scary, and there's so much to do, right? It kind of holds your hand through the basics of that. But what we hope is that, by showing you how you can solve some of these scientific problems using these skill sets, will actually inspire students to think of their own problems that they want to solve. And so if you have these skill sets and you develop them at home, there are a lot of public resources available. There are public genetic data sets available, for example. You could take a data set, for example, from patients who do not have cancer, take a data set from patients who do have cancer, and you can learn some of these basics of coding, figure out how to compare them, find similarities and differences between them. We hope that will inspire people to start to ask some of these questions at home and think about the own type of scientific questions that they're really interested in looking into professionally later on, or non-professionally, just for fun at home.
Right. Amazing. And thank you guys for creating that resource. Like, that's awesome and so useful for people to be able to kind of start exploring that at a younger age and to be able to use that in their future research.
In the second part of this podcast interview, Dr. Davis mentions that ATP is an energy molecule that our mitochondria pump out. She also mentions that the first 2 years of a PhD are often spent taking classes and then going into lab after. After the second year, a lot of time is spent in lab depending on what you get a PhD in. You also have to focus on communicating through writing and making figures. A plasmid is a vector that a gene of interest can be inserted into so that it can be put into live cells, which give the cells the instruction to make the specified gene. Lastly, Outside of working in the lab, Dr. Davis has taught classes, gotten involved in science communication groups, and studies science policy, such as how policy regulates science and how science can inform policy.
And
We hope so.
so now say, you're in graduate school and then you graduate, you get your PhD or whatever other degree that you're going for. What do you do after that? So you have your PhD. Do you have to go into a field like that you got your PhD in, or can you kind of go if you figure out you like something else, you can research that? What do people usually do after?
Great question. So, like I was saying earlier, I mean, the main thing that you do as a scientist is you really develop your research skills, right? You really figure out how to ask good questions, look at information, and draw conclusions from it. That skill set allows you to do so many different things, right? So I have honed that skill set by studying something really particular, like how mitochondria move in neurons, right? I don't plan to do that for the rest of my life, even though I think it's a perfectly interesting and valid career choice. I can take these skill sets that I've gained in the lab, asking these questions, to do all kinds of things. So personally, I'm pursuing a career in science policy. I'm really interested in looking at big questions like public health and science education, and how our policy can regulate and make things better for the next generation of scientists who want to go into scientific training like myself. But there are so many other options. So I'll just name a couple. So you can go into academia. You can become a professor on a tenure track. You can go into academia and you can teach at a university full time and maybe not do as much of the research side, or do half and half. You can go straight into a pharmaceutical or biotech company. So maybe you really love the research part of it, but you don't really like teaching or the academic part of it or the writing part of it, and you just want to get to do the really cool science experiments. And so pharmaceutical research, biotech research is a great way to do that. I actually have several friends and colleagues who have taken their graduate student research. They've turned that into companies. They've started their own companies researching questions that they're excited about. That is an option as well. I have friends who go into consulting. So that's like, I think of consulting as like professional problem solving, essentially. A company or a nonprofit comes to you with a problem that they have. You're a really good critical thinker. And so you can kind of look at this problem from the outside and you can give them your advice. You can do research on how to solve this problem. You can think about the best way to go about solving the problem financially and give them recommendations on that. So that's a path that you can go into. So many different options, really. Some people go into investment banking after they finish their PhDs.
Mm-hmm.
So the sky is truly the limit, and it's really just the limit of your imagination. And also, I've watched people make incredible or find incredible ways to blend their interests together as well. It doesn't necessarily have to be, this one niche, right? People can blend their interest for art and teaching, for example, and turn that into a career. So just so many options that you can go into with a STEM degree.
Awesome. And then another question I had was that you mentioned that neurons can be really long, like up to 1 meter long sometimes, and then mitochondria move around in neurons. So I'm kind of wondering, how big are mitochondria? Is there like a general size?
Yeah, they're quite small, actually. So the scale that we look at them are on micrometers, so micrometers. And they're under the size of 1 micrometer, which is very, very small. They're just these, it's actually, I'm going to take that back a little bit because it's a complicated question, because mitochondria are different sizes and different cell types. And they can also do this really cool thing. So you have a single mitochondria. They can do this thing called fizzing and fusing, which they do constantly. So mitochondria will find another mitochondria and it will fizz to it and it will become a monster long mitochondria, or sorry, fuse to it. It becomes a monster long mitochondria. And then it can fizz where it separates into these smaller little mitochondria chunks. So actually, I'm going to take back the size because they constantly change and it depends on the health of the cell. It depends on the cell type, but that's around the scale that we're looking at them on, is like this micrometer size. And so whenever I'm measuring them, so I'll have like my picture of my cell and I'll put up a little scale bar, like a little ruler, and it'll have like 10 micrometers on it for my cell. If I zoom in for just my mitochondria, then I have like a one micrometer scale bar to kind of show you where they're at around that range. But they're constantly changing from tens of micrometers in length to just one, and sometimes get even smaller than that.
Right. Okay. Awesome.
It's never an easy answer with science.
I feel like that's science in general. It's never, it feels like you can never just say this is the answer, because things are constantly changing around you.
Things are constantly changing.
And then could you kind of give some words of encouragement, if you have like anything specific that you would say to people that want to go into science? Like, you kind of laid the foundation. These are some things that maybe you could do if you want to go into science to be able to get into it. And then do you have any words of encouragement for those people?
Yeah. So, I mean, my biggest recommendation is to just keep going. I think one of the most important things that you can have as a scientist is grit. And whenever I think of grit, it's really just like moving forward against all else, whether somebody tells you you're not going to be a good scientist, or, maybe you didn't get into that summer internship, or, you didn't do so well on your last exam. Just keep going forward and keep looking for new opportunities that are going to allow you to get hands-on practice with science. I will say, during my my undergraduate, I didn't have perfect grades. I went to Harvard University for graduate school, right? A lot of people think, oh, you must have 4.0 GPA. You must be an amazing student. I didn't have perfect grades. I did terrible in my college calculus classes, like really bad in them. I'm not good at math. And I think, what really made me competitive for applying to graduate school was that I just had this continuous demonstrated interest on my resume and evidence in my personal statement, where I just kept pursuing opportunities to do science. And I was constantly in the lab during the year. I was pursuing things during the summer. I was always looking for the next opportunity in science. And I think that demonstration of, my passion and interest there really came through. And it also, that skill set that I developed in undergraduate became so important for graduate school, because graduate school can be really hard, and it can also be really critical. A lot of what we do in graduate school is, we're performing either in class or in the lab or in front of a committee of our advisors, and they're telling us their most critical thoughts about how we're doing and the work that we're doing, right? And so you really have to kind of develop, a tough personality, and just kind of take on the attitude that, yep, I'll take this criticism to heart, I'll think about it, but I'm going to keep moving forward towards my goal, and not letting those criticisms really get to you or stop you. And I'll be honest, I had people tell me, maybe I shouldn't pursue a PhD in science. If I was really interested in science policy, if I really like teaching, maybe I should do those things. Why didn't I need a PhD in science? Mm-hmm. And I said, I really want a PhD in science, even though I'm going to do those things. I'm going to do both of them. Yep. And so just not letting anyone, tell you how to do things, or tell you, that that you can't do it, I think is my biggest piece of advice. If you want to do it, just keep moving forward, and eventually you're going to do it.
Awesome. And then, of course, can't get enough of the science. So I was hoping to close out on just one more science fact. So you kind of talked to us a little bit about, like you said that you did a short thing in a virology lab at Johns Hopkins for your undergraduate, like an REU there. And then you also mentioned something about RNA and viruses. So could you kind of like just give us some virus fun facts, I guess?
Oh,
I mean, fun,
okay. Well, I'm not a virologist, so this is hard. Let me take a drink of water and think about it. Virus fun facts. You know, let me think. Viruses are really, really fascinating. The funny thing about becoming a scientist is you kind of like, I think you become aware of like, whenever you become a specialist in something like mitochondria, I become hyper aware of how much I don't know about viruses. Right. And so right. It makes me nervous to like say anything like that. But you know, I will just say viruses are super cool. There are so many that we don't think about. So like, you know, we think about like regularly, okay, so all of a sudden, you know, COVID is on our minds. We probably never really thought about coronaviruses before, even though we all get coronaviruses every year. Like that's our common cold. And the flu virus as well. Ebola is one that we've like heard about recently. I think the craziest thing, you know, for me, and especially in the last year, that's become really obvious about or interesting about viruses, and I've started to think about more, is how they evolve and change over time, and how cool that is. And so, like, whenever we talk about, you know, COVID, for example, a lot of scientists believe that COVID was in an animal and evolved there, and then was able to transmit to humans, right? Mm-hmm. And so something that I've become kind of hyper aware of, you know, someone being interested in policy and public health and this kind of thing, is that there are actually people all over the country who follow these viruses and will like take samples from animals and humans in the clinic and sequence them to see how they're evolving. And it's become a really important way, I think it's going to be a really important way in the future, for us to follow the evolution of viruses that could potentially become dangerous for us, like COVID was, where it evolves into like epidemic or pandemic spread. We can actually study these things and get an idea that they might be coming before everybody has it, right? And so that is like part of virology to me that is most interesting right now, is not so much the viruses themselves, but like how can we get ahead of them? Because we know that they're going to evolve. We know that they're going to mutate. We've like seen all these different strains of COVID that are going around. Mm-hmm. And so what is the best way that us as scientists can plan for them and like set up surveillance of viruses to figure out when the next dangerous virus is coming? Mm-hmm. Whether it's an Ebola virus or a flu virus or coronavirus or who knows. That is something that I think is really cool. And I'll just add in one other really cool thing that people are doing with viruses right now, if you're interested in thinking about viruses. People are using viruses as vectors for therapeutics.
Wow.
And that to me is so cool. So basically they can take, like, you know, viruses are really good at invading our bodies and getting into our bodies, right? So people can take viruses and they can engineer them so that the disease components aren't there anymore. And then they can give these viruses helpful things. And so if you're interested in thinking about viruses, one thing that I would encourage people, especially at the undergraduate stage of their career, is to look at viral vectors for therapeutics. Because I think that it's going to be a really exciting future area of science. And there are already therapeutics that are, you know, approved and on the market for these kind of things. So we're going to start seeing a lot more of this coming up. So that is just like, I'll just kind of plant that seed in people's brains, is that that's going to be a really exciting area of upcoming research utilizing viruses that I think we're going to start hearing a lot more about.
All right. Very cool. And I just want to say thank you so much for joining us today and for being willing to come on and share some really cool science facts with us and share about your journey and advice. That this was very helpful, I think.
Good. I hope it's helpful. And thank you so much for having me, Lizzie. This was a lot of fun.
Yes, of course.
In the last part of this podcast interview, Dr. Davis mentioned that the skills we learn as scientists can be easily transferred to other careers. So if you find out you're interested in other things, know that this is normal and totally possible to go into these other things that you're interested in. She also mentions that you can go into academia, teach as a professor, go into a biotech company, consult for other people, or even do investment banking with a PhD. She mentioned that mitochondria can go through the process of fusion and fission. Fusion is where they combine together, and fission is where they break into smaller pieces. Mitochondria are looked at on the scale of micrometers, which is 10 to the negative 6. They are different sizes in different cell types. She mentions that one of the most important things to have as a scientist is grit. Regardless of how you have done in the past, keep going on. It shows schools your interest by continuing research and looking for opportunities. Lastly, Dr. Davis mentions that we can track how viruses are evolving by taking samples from animals. Viruses are also used as vectors for therapeutics. Thank you for watching this episode of the Doc Talk with Liz podcast. Don't forget to like and subscribe for future videos.